Doing my past 6 or 7 years, kind of exhausted, but we'll work through the lecture today. In addition, had to sit through the 2 hours of that Pepsi show last night because my wife was there. Needless to say, since I'm here today, we did not win the $1 million nor the $1 billion. She just had a good trip, I guess. So we'll get back to the embryology. I'll play a video first, and then we will pick up with Lecture 7 and push on through Lecture 8. So here is a video showing the germ disc and the ectoderm, the mesoderm just beginning to form, and the endoderm with the yolk sac side and the amniotic sac side, primitive node and the primitive streak. So we'll see the changes that occur over time. So there's a cross-sectional plane through that area with a plate-like movement of cells pushing along the epiblast layer to the primitive streak. And you can see the movement of the new cells migrating and interposing in between the surface cells on the dorsum and the amniotic sac side, and on the gut side, if you will, the yolk sac side. And these are the mesodermal cells. Thereby, we now have three-layered embryo, three-layered germ disc, the embryo. So with this development, as we just followed, there's the additional event at the primitive node that we had mentioned before, and we will continue to emphasize this and look at it in a bit more detail. And this is the solid core of cells that will migrate from the primitive node in between the ectoderm and the endoderm. We'll refer to them by that name now, toward the prechordal or prochordal plate. Both names seem to be used interchangeably. And as the notochord process moves forward, we said that it is a placeholder, but it also is a stimulus for transition of the ectodermal cells immediately overlying it, and that they begin to become taller, and will divide and increase in number and spread laterally and form what is called the neural plate, which eventually will crumple, and form the neural fold, with the midline being delineated by that placeholder, the notochord. So we've been talking about symmetry, left and right, and then asymmetry, rostral from caudal, and we can see already that there is an asymmetry from rostral to caudal of the developing neural plate and neural fold. We see also that the prechordal plate is beginning to undergo transition and now is being referred to as the oropharyngeal membrane. The prechordal plate showed up very early on in the two-cell layer stage, and we said that it will eventually become a portion of the oral cavity. So this then is referring to the neurulation, and now we can talk about the gastrulation. The three germ cell layers, as we said, are all derived from the epiblast cells. Originally, the mesoderm, as we had indicated and just viewed on the video, is going to form different portions once it migrates away from the midline. There will be paraxial, intermediate, and a lateral plate of mesoderm. So I guess I should emphasize that the previous slide was making that point again, that the nervous system is one of the structures or organ systems, the central nervous system, that will originate from the ectoderm. So this is our exercise right now. We're trying to make the point of example, not exhaustive listing, but of example of the derivatives of these germ cell layers. So the nervous system was our one example of the ectodermal derivative. Now we can talk about mesodermal derivative as a result of this gastrulation. So the mesoderm is going to give rise to smooth and skeletal muscle and actually cardiac muscle. So all of the muscle originates from mesoderm. All of the connective tissue, including the blood and the bone. There might be slight exceptions to these, and we'll make those points as we go along, but these generalizations are quite safe at this time at least. So the blood vessels and the cardiovascular system, as I said, the cardiac muscle. And then the reproductive and the urinary systems. But we need to make the point now that these are the muscular and the connective tissue and the vascular components of these organ systems. The epithelial lining, such as the endometrium, will originate from endodermal structures, as we will see. So all of the linings of the organs of the respiratory and the gastrointestinal systems will originate from the endoderm layer. So this is figure 5.5 from our text, and we see that the inner cell mass is shown here, the epiblast contributing to the trilaminar embryonic disc with the ectoderm and its derivatives, and the mesoderm and its derivatives, and the endoderm and its derivatives. So just quickly to go through these, the ectoderm giving rise to surface ectoderm like the epidermis, the hair, nails, cutaneous and mammary glands, anterior part of the pituitary gland, the enamel of the teeth, the interior of the ear, the lens of the eye, and then the neural ectoderm that we saw, that neural plate forming, will give rise to a neural crest, which will form all of the cranial and spinal sensory ganglia and nerves. So basically the peripheral nervous system in general terms. Again, there's specifics and exceptions that we make along the way. Medulla of the suprarenal or adrenal gland, certain pigment cells, the pharyngeal arch cartilages. So here's an example of connective tissue coming from neuroectoderm and not from mesoderm. The head mesenchyme and connective tissue, another example. The bulbar and coronal ridges of the heart. So other examples of exceptions to the rule. The neural tube of the central nervous system will give rise to the central nervous system, the retina, the pineal body, and the posterior part of the pituitary gland. And the mesoderm will form the lateral, intermediate, paraxial, and the head mesoderm, as we had indicated. And the lateral is going to become connective tissue and muscle of the viscera. So we'll follow this as it moves out laterally and then comes in to become a component of the gastrointestinal system. The serous membranes of the pleura and the thoracic cavity around the heart, the pericardium, and around all the abdominal structures. The peritoneum, remember the peritoneum on the uterus was the perimetrium. So the perimetrium originates from lateral mesoderm. It'll give rise to the primitive heart, the blood and lymph cells, the spleen, and the cortex of the adrenal gland. The intermediate mesoderm will be giving rise to the urogenital system, including the gonads, the ducts, and the accessory glands. And the paraxial, very close to the axis of the organism, will give rise to muscles of the head, the striated skeletal muscle, the trunks, limbs, the skeleton except the skull. It'll give rise to the dermis of the skin and other various connective tissue. The head mesoderm will give rise to the skull, the connective tissue of the head, and the dentin. And then the endoderm, there will be epithelial parts to line the trachea, the bronchi, and the lungs, the epithelium of the gastrointestinal tract, the liver, the pancreas, the urinary bladder, and the urachus. Now, these will be only either the parenchymal cells, like of the liver and the pancreas, or the epithelial cells of the urinary bladder, the smooth muscle and things like that are going to come from mesoderm, and the vasculature from mesoderm, the neural innervation from the neural crest of the ectoderm. And then there will be epithelial parts of the pharynx, the thyroid, the tympanic cavity, the paratympanic cavity, the tonsils, and the parathyroid glands. So the challenge is, of course, to familiarize yourself with the anatomy of all these structures and beyond memorizing terminology and lists, trying to put them into place. So from which germ disc does the blood, the lens of the eye, the epithelium of the kidney tubules, and the thymus develop? The grammar on this is not the greatest. So what we see, again, is the question with the answers provided. The blood is coming from the mesoderm, so there is no single answer to this question. The lens of the eye is coming from the ectoderm, the kidney tubules are coming from the mesoderm, and the thymus from the endoderm. So we see another exception, the epithelium of the kidney tubules coming from mesoderm. So there's always these general rules and then the exceptions, and part of the labor of this class as an upper division class is to become familiar with and try to remember all of the exceptions as well as the generalizations. So from which germ disc does the bone, hypophysis, parathyroids, and thyroid glands develop? The bone is from mesoderm. Hypophysis, which is the pituitary gland, comes from the ectoderm. The parathyroid gland from the endoderm. The thyroid gland also from the endoderm. So you have to imagine how these can be converted into questions. Which of the two following adult structures are derivatives of endoderm? So it'd have to be parathyroid and thyroid gland for that statement, that particular question. So now we can talk a bit more about the notochord. There is the proliferation. They are specialized cells. They do extend cranially from the primitive streak toward the precordial plate. They are basically mesodermal cells. They're the precursor of the vertebral column. They're going to initiate those events in the nearby mesoderm to form the connective tissue, which includes the bone, of course, of the vertebral column. They will also interact with the neural fold that will become the neural tube, such that the vertebral column, of course, surrounds the spinal cord as we are familiar with it. Persist, as I noted before, is the nucleus pulposus of the intervertebral disc. It induces overlying ectoderm to form the neural plate, the CNS, as we saw in one of the previous slides. So we now can follow figure 4.7 from the textbook to see how this notochord develops. So in panel A, we are looking at a dorsal view of the embryonic disc about day 16. And again, we see the cut edge, the blue, the cut edge of the amnion, so we know we're looking at the dorsal surface. We see the primitive pit in the primitive node with the primitive streak back here. We now see that a cloacal membrane is identified. So this is, again, we said we established with the precordial plate and the primitive streak, pit, and node, a rostral toward the head and caudal toward the tail. And the cloaca is going to be the primitive pelvic area, if you will, that will become the pelvic visceral structures for the gastrointestinal and the urinary systems, basically collectively. Any of you that are familiar with the anatomy of chickens know that there is a common opening, or even birds, a common opening, the cloaca, into which both urinary and gastrointestinal wastes are excreted. So we follow to panel B, and we see that at the primitive pit, some cells forming mesoderm a solid core, the notochordal process are interposing themselves between endoderm and ectoderm and move toward the prechordal plate. This is the notochord. There is a short canal at the primitive pit called the notochordal canal, and we'll see that at one point does form an opening between the amniotic cavity, a transient one, between the amniotic cavity and the yolk sac, but at this time it's not a focus of our attention. So from a sagittal view, we can see that notochord pushing into the mesoderm with a canal in there, and it is pushing toward the prechordal plate. In a cross-section, transverse section, as in panel D, we see the notochordal plate as we process with the canal inside of it, as we had seen before. And also we see the thickening of the neural plate as an induction from the notochord. And this, of course, continues. C and E are about 18 days, and the D and the F are the transverse sections through this area. So by about day 18, the notochord has completed its migration toward the prochordal plate. As we proceed then to figure 4.8, then we're going to again take a look at the dorsal view. The notochordal process has pushed through. The neural ectoderm is just beginning to react. The surface ectoderm, which is triggered to become the neural ectoderm, is reacting, and the cells are becoming tall columnar cells just above the, I guess I should do panel B first, just dorsal to the notochord. And we see the the notochordal canal extending into the notochord. So panel C is showing us that the notochord reaches the prochordal plate, and then there's the localized thickening of the neural plate at its rostralmost extent, and the region in just rostral to this is called the oropharyngeal membrane. When we see a transverse section of this stage in panel C, just shortly after or around 18 days, we see that the notochord now has opened up and is just allowing the continuity, this transient continuity, through what's now called the neuroenteric canal between the amniotic space and the yolk sac. In panel E, we're seeing the levels of the section G and section F, so we can now take a look at this area here of F, and we see that the notochord is only partially open. It's actually forming kind of an omega type figure here. And then farther rostrally, it is still the solid layer of cells with at the circumference of and surrounding the neural canal, the notochordal canal. And all along the way, we see the reaction of the surface ectoderm to become taller cells, tall columnar cells, and begin differentiating into the neural ectoderm. In this panel G, we also see some of the mesoderm that has gone on to differentiate or cluster, let's say, not necessarily so much differentiate, into the paraxial mesoderm, the intermediate mesoderm just lateral to that, and then farthest lateral, the lateral mesoderm. So we're introducing that idea in these contexts as well. So what is the notochord in the primitive pit? Indicate its place in a schematic drawing showing the mid-sagittal section of the 16-day-old embryo. So you will not be asked to draw, but you may be asked to label. So at the cephalic end of the primitive streak, the primitive node develops. Primitive pit develops from the primitive node by cell invagination. So this is a new term that we are learning. The cells will push inward and create a sheath, so it is invagination. So these cells migrate straight forward towards the cephalic end and form a tube-like process, the notochordal or head process. And the canal of the head process is the cranial extension of the primitive pit, and the dorsal wall of the notochordal process proliferates and forms the definitive notochord. So it's a slight variation of my statement of a solid core of cells. It actually is a rim of cells with a canal down through the center. So here we should be able to label these now. Number 1 certainly is mesoderm. Number 2 certainly is notochord. Number 3 certainly is ectoderm, and we see the thickening of the neural ectoderm in response to the presence of the notochord. Number 4 is entoderm or endoderm, and then 5 would be the yolk sac. So you should be very familiar with this type of illustration. Here we see immediately the primitive streak, number 3, tells us caudal end, prochordal plate, and of course there is this pear shape with the narrowest portion of it being caudal and the greater diameter being rostral, but still the primitive node with the primitive streak extending caudally is very important, the notochord extending toward the prochordal plate being our major points of orientation. So the new thing identified now, of course, is the future cloacal membrane. And number 1, the prochordal plate is going to become the oral pharyngeal membrane. So we now have another illustration, so plenty of things to label. The amniotic cavity should be very apparent to you now, not so much from any other information other than knowing that this is the tall columnar cells of the neural ectoderm. So dorsal to that would be the amniotic cavity. So number 2 are the ectoderm, the epiblast cells. We're in that transition of terminology as the notochordal process with the notochordal canal can be seen here. Number 3 is the primitive pit that leads into that notochordal canal. Number 4 is, of course, the prochordal plate, going to become the oropharyngeal membrane. And number 5, the 5 disappeared for some reason, is the notochordal process. And number 6, of course, would be the primitive streak extending caudally from the primitive node and pit. So with gastrulation then, the oropharyngeal membrane is forming, and it is the site of the epi and hypoblast cells fused together. So what this means is they fuse together, form tight junctions, other types of intracellular junctions, and they're going to prevent any cells from interposing in between them. And this is very important for the formation of these membranes, because these membranes will deteriorate eventually or perforate to form the opening from the oral cavity into the oral pharynx, and of course to create the opening at the anus. So you don't want any mesodermal cells interposed to form a thicker layer of connective tissue and make it difficult to perforate these structures when they must become patent or open. This also is going to be a key to tell us where the cardiogenic area is. So our heart forms in front of our head very early on and then flips, as we will see, down into the chest cavity. So the oropharyngeal membrane at that prochordal plate is telling us that nearby is going to be the cardiogenic area. So everything is about establishing polarity, either symmetry or asymmetry of the system. The cloacal membrane will be the anus, and again, it is the fusion of the epi and the hypoblast. We also saw in the previous illustration the allantose, or it is a diverticulum of the yolk sac, in which the early blood cells are going to develop, and it eventually contributes to the formation of the bladder. So on day 16, we see that coming up here. So number 7 would be the allantose. So this number 8 is the the yolk sac, the endoderm lining the yolk sac, and the diverticulum of that, a little pouching, pushes up into the connecting stalk shown by this shaded area, and that will be the allantoic stalk and eventually the allantois, and it will eventually develop into the urinary bladder and this other structure called the urachus that we'll illustrate a bit later. So the difference between the extraembryonic and the intraembryonic mesoderm, where do they originate from? So the extraembryonic is from the trophoblast, and they form a fine, loose connective tissue which is located outside the embryo. So we had talked about those two. There was the somatic, and who's going to help me? What was the other one? Splanchnic. There was a somatic and a splanchnic extraembryonic mesoderm that we saw in a previous lecture. Now the intraembryonic mesoderm that we introduced is the third germ layer between the epiblast and the hypoblast, and it originates from the epiblast layer. And it will establish a paraxial, intermediate, and lateral intraembryonic mesodermal clustering, if we want to think of it that way. So everything is about migrations of cells, either as plates or as clusters or as individual cells crawling around and then collecting together to form different structures. Prime example is adrenal cortex and adrenal medulla, as we will see later in the semester. So what are the components of the extraembryonic mesoderm? Well, here they are listed for us. So there is a somatopleuric mesoderm and a splanchnopluric mesoderm. We're now seeing this pleuric added to the somatic and the splanchno, and pleuric should automatically make us think of the thoracic cavity or the chest cavity for the pleura, the lining of the lungs. It'll also extend into the connecting stalk to contribute to the connective tissue of the umbilical cord, and then the chorionic mesoderm that will help to give rise to some of the chorionic vascular structures. So the connecting stalk, what is it and how does it develop further? So it's part of that extraembryonic mesoderm that I said. It contains the allantois. We'll see a vitelline duct, and the umbilical and the vitelline vessels will develop within that. And then later the amnion will envelop that connecting stalk and the yolk sac stalk with all of the vessels, of course, forming the primitive umbilical cord. Now as we proceed to lecture number 8, we can follow a bit more closely the events of neurulation and begin thinking of the folding. So we were talking about this very metabolically active cluster of cells, the conceptus. Now it is an embryo, as we are well into the 3rd week, and we have cells dividing and migrating and moving all over, and we still are keeping in mind that purse string effect. So we get rippling and crumpling. So there's folds and ridges and all types of things like that being formed by the cells in their movements. So there is not a tremendous amount of space to move laterally, so we are going to see this folding as a result of the plate-like movement of cells in the formation of the ectoderm, endoderm, and mostly ectoderm and mesoderm, because they're going to squeeze around the endodermal part. So this will contribute to the formation of the body cavities, and as well, we know we have to establish the circulatory system. It's getting, the embryo is now big enough, and we know that the chorion is getting quite well developed, and the blood lakes are there. The extraembryonic mesoderm is out in that area. We're ready to create the embryonic side of the circulatory system. So we start first with the neural tube, the neurulation. So it is beginning during week 3. We already went exhaustively through the influence of the notochord, to create first the neural plate. So this is the thickening. We get the tall columnar cells. Then they're getting tall, plus there's not enough room for the lateral expansion of everything. So we get a bit of a crumpling and folding. So there will be a fold in the center, just dorsal to the notochord, and then on either side, there will be a groove in the center, just dorsal to the notochord, and on either side will be the fold, that crumpling. So the groove will be the central invagination along the long axis of the neural plate from rostral to caudal, again consistent with the notochord, placeholder, and it's bordered by those neural folds. Eventually this will meet, the crumples will come up around and they'll meet each other and fuse together. So those folds are going to fuse together and form the neural tube. And this process will be completed by the end of the 4th week. So we're now proceeding from 3rd week to 4th week. And in the 4th week, we're already starting to have not only cytogenesis and histogenesis, the tissues, but we're beginning to see some of the early signs of organogenesis. The central nervous system and peripheral nervous systems are beginning to appear. Again, the circulatory system is not too far behind, but the neural system is going to be one of the first ones to begin to develop. Circulatory is most important because we need to get the nutrients and waste moving around, but we have to get the nervous system set up so that we can enclose it within the organism and get it on its way to becoming established. In other words, to make connections with the structure that's going to innervate, and then those contacts will stay and just the cables will get extended. So we have to think now, as we see the nervous system beginning to appear, that it is appearing early, before the heart is there, before all these other things are there, because it's got to get ready and cause differentiation of some neurons so they can send out their axons and make connections with the innervated structures, and then the axons will just continue to get longer as those structures migrate or move around to their final destination. So that's an important concept to put in our mind. I just happened to catch a bit of a show on stem cell research for recovery of spinal cord transections, and there's still this idea that we can accomplish this 100%. It will be biology basically at its best when this does happen, because what we are asking of the nervous system is to do something that it doesn't do developmentally. We're asking for it to reestablish a connection that it lost over a great distance. Remember, these connections established very early on over very short distances and just grow in length with the further development. So it's a very important concept to have in mind. And then when you study the anatomy of the adult structures, for example, the vagus nerve is not named the vagus because it wanders all through the chest and the abdominal cavity. Makes its connections very early in the throat region, and then as things move away, it just gets longer. So it is not this type of a thing where something develops on the other side of the room and suddenly you're a neuron and you got to send out a process. Just looking around, you happen to bump into it and you say, oh, am I supposed to innervate you? Oh, okay, now I'll innervate you. You do that very early, and then as that structure moves to the other side of the room, you go right along with it. So this creates a challenge in replacing those connections if they are lost through injury or disease. So this is why it is so important. So we are brought back then to this view, primarily panels D, F, and G, showing the neural groove and the neural folds on each side, formed both by the thickening of the epithelium and by the crumpling. So this is an illustration from the Grant's anatomy again, and it is showing the cut margin of the amnion. We see the yolk sac extending inferiorly. Whenever we see amnion, we know we're looking at dorsal surface. If we're looking at dorsal surface, and we already have the neuroenteric canal, or the primitive node in the pit, and the neural groove extending rostrally from that, that we are looking at the embryo. Safe to say that it's the embryo. And here's the primitive streak coming around, so the cloacal membrane will be back here, and we can see the body stalk connected to the developing cytotrophoblast layer that is pushing out into the syncytiotrophoblast layer to form the chorionic villi. So now we can transition to figure 4.9 from our textbook, and again, the blue color kind of gives it away, but we have primitive node with the pit, primitive streak, and the rostral in this direction, and we can see the folds are developing with the groove here. So we know we're looking at the dorsal surface with the amniotic sac cut edge shown here. We are beginning to see some spaces showing up way out here. These are coelomic spaces. So these are the first signs of body cavity formation. With a high magnification of a transverse section, as indicated here, the level of section B, just at the caudalmost end of this neural fold. We can see in the neural groove, the underlying notochord with the notochordal canal, and the folds on either side, pushing on over to the connection of the amnion and the associated mesoderm, somatic mesoderm. So the somatic mesoderm and the splanchnic mesoderm come together here as the extraembryonic, and we see the intraembryonic forming paraxial, intermediate, and lateral. The lateral mesoderm is where these coelomic spaces are beginning to show up. These are going to become parts of the body cavities, as we will see. And this view, panel C, is showing that those coelomic spaces in that mesoderm, and we're now going to look at this level of transverse section D, where we now are seeing evidence of the first somite, and this is going to again give polarity, not so much polarity as it is to begin to establish symmetry in the sense of segmentation. Now we're going to see segmentation. So we've all seen earthworms, and we know how they're segmented. So as ontogeny recapitulates phylogeny, so ontogeny is basically what we're studying. We can see, we've already seen reference to chickens and birds, and now we're starting to talk about worms, so we are seeing that indeed ontogeny does recapitulate phylogeny in some sense. So there is a segmentation that is going to occur. So here we see the paraxial mesoderm in this area about the mid-level rostodocaudal of the embryo is where the first somite is going to appear. So the first somite is going to appear in the middle, and then they're going to begin to develop rostrally and caudally to that. And at this stage, the intraembryonic coelom is beginning to become a continuous space. So this lateral mesoderm is now differentiating into an intraembryonic somatic mesoderm, going to line the body wall, and then the intraembryonic splanchnic mesoderm that is going to line the yolk sac for now, but it's going to become the gut tube, basically. So we're now beginning to see the very early formation of the body cavities, in this case, what's going to become ultimately the abdominal cavity, but it'll also extend its abdominal pelvic, and it will be continuous, as we will see, with the pleural cavity. So again, how do we create this type of a space? The cells nearby are going to secrete or allow the accumulation of extracellular fluid, interstitial fluid, and the cells will let go of each other. They won't have tight junctions, and so that fluid will accumulate, and you have a body cavity, a potential anatomical space with just a little bit of fluid accumulated there. And of course, we have many examples of those in the adult: pericardium, the pleura, the peritoneal, the bursa at our joints, the joint cavities. So it's a very common thing for our cells and tissues to do. Now this view is beginning to show the body cavities, that extraembryonic coelomic space now is becoming continuous. So we're not following the establishment of the circulatory system right now, but we see a pattern emerging. Form these little collections of extracellular fluid. They are lined by cells. In this case, the cells lining are not going to become blood vessels. They're going to become mesothelial or serosal membranes, mesothelial cells that contribute to the formation of serosal membranes. And these serosal membranes are what line our body cavities. And so we see that there is a continuity from the pericardial region. So remember, the prochordal plate was going to be up here. Just in front of it would be the oropharyngeal membrane. And this told us that this is where the heart is going to develop. So the pericardial coelom is going to form around that area. And this will extend all the way into what would be the chest cavity ultimately. So there is a pericardio-peritoneal canal that's going to extend all the way from where the heart would be, through where the chest will be, and all the way into the peritoneal cavity where the peritoneal coelom is already forming. So we now notice that there are two additional, on each side, left and right, two additional segments, the somite in front of, or rostral, and behind, or caudal to that original somite. So we are now at the three-somite stage. So there are different ways of referring to the stages of development now, especially of the embryo. One that we can use is, other than, of course, the days from conception, we can refer to it as somite stage. So it's the one somite stage and now the three somite stage. And again, this same section, transverse section, is showing a similar pattern of the intraembryonic coelom, and we see the splanchnopleur forming here, and the somatopleur forming on the ectodermal side. So the body portion and the gut tube portion. Most importantly, what we begin to note now is the dramatic change in the structures that are related to production of the central nervous system. So the edges of the neural folds are approximating each other, and very soon they will connect and create associations with each other so that they stick to each other more than they do the nearby surface ectoderm, and so they'll grab onto each other and be pushed underneath the surface ectoderm. They'll let go of the surface ectoderm, and then the two sides of the surface ectoderm, right and left, and you can't see any of my hands, not that it would matter, but anyway, the two sides of the surface ectoderm are going to have to come together and talk to each other and say, we need to fuse shut and let go of the nervous system so it can be submerged and surrounded by mesenchyme, mesoderm to form the vertebral column and so forth that will contain the CNS. So many events occurring for this to happen. It's not all just physical displacement. It's cells communicating to each other, creating connections, letting go of connections, and combining that with the physical forces that are occurring. So we see this special event about to occur where the neural folds will come together. So which of the following is the correct timing of appearance of the structures of the central nervous system? Groove, tube, plate, definitely not wrong. Plate, groove, tube, sounds right from what we just saw. We get the local thickening, and then the groove as we get a crumple and a ripple in there, and then the ripple becomes the full wave, the crests of the waves meet each other and form and close off the groove to form the tube. What is the neural plate? What is meant by neural induction? So the plate is the thickening of the surface ectoderm. It's the primordium of the nervous system, and the notochord is what is inducing this surface ectoderm that results in the formation of the neural plate. So that is the neural induction. So there's different types of induction, obviously. So this is the end of our time for today's presentation. So I will stop with this slide in Lecture 8, and we will push on. So we continue to push on. A reminder, Exam 1 is 1 week from today. So I will begin efforts, now that I've got this one big job out of the way, the review of my life for the past 6 or 7 years, as I said, we'll shift over. We'll get the missing lectures taped and posted, and we'll get the exam posted so that it will be available for you to take in class next Monday. And if there are any complications with administering all or part of it through the WebCT in class on Monday, then we'll let you know, and we may show up with something in print. So I appreciate your time and attention. I'll see you on Wednesday.
Dr. Felix?
Yeah. Oh.
I finished up the material I wanted to do for the bonus questions like we discussed. I couldn't for the life of me figure out how to put it up on WebCT so that it was accessible.